Professor Marcelo, I request you to please stop the sharing of your slides so that like we can start. Yeah. Okay. Thank you so much.
Can we play the video?
Yeah. Devendra sir, should I start with the video?
हाँ प्रतिभा करो स्टार्ट.
I think, are you all able to see the video?
No. Not really.
Not really.
Pratibha ji.
Okay.
Yeah.
Oh, I think there may be some problem. I should stop sharing now. Or we can straight away, there may be some problem. Let me check.
I think Palak can start, Pratibha ji. Are you ready, Palak?
Yeah, ma'am, sure.
Pratibha ji, what do you say? Shall Palak start?
Yeah, she can start, I think. Or I can share from the link if you want.
A very good afternoon to all. So am I audible and visible to everybody?
Very much.
Yes,
Very much,
you are.
Okay, thank you. Starting with few Spanish lines. Los ganadores nunca. Abandonan de los que abandonan nunca ganan. This simply means winners never quit. And those who give up never win. A very exuberant afternoon to one and all present here. We all were, and even now, are dealing with the pandemic, but that hasn't stopped our limits to learn and grow, isn't it? In such time, webinars come into play. It is a platform through which we seek knowledge through online interaction. I, Palak Pal, second year student of Industrial Chemistry, Deshbandhu College, warmly welcomes the respected teachers and my dear friends on behalf of the Chemistry Department. I would like to respectfully and cordially welcome respected Sir Professor Rajiv Agarwal, the Principal of Deshbandhu College, Sir Dr. Deonandan Kumar, the Teacher in charge of Department of Chemistry, Sir Dr. I.K. Singh, Coordinator, DBT Star College Scheme, Sir Dr. Aditya Saxena, Coordinator IQAC, and Ma'am Dr. Pratibha Kumari, Program Convener, without whom we couldn't have organized the webinar. We are honored to have such an exemplary speaker who was highlighted as emerging investigator by the Royal Society of Chemistry Journals Chemical Communications 2015 and Journal of Material Chemistry B 2017, and a lot more achievements. None other than Sir Professor Marcelo Calderon, the University of the Basque Country, Donostia, San Sebastian, Spain. Before starting today's event, we will virtually have traditional ceremony of lamp lighting and Saraswati Vandana as a tribute to Mother Saraswati, the goddess of knowledge. soothing. And now we all are sanctified. Let's proceed further onto the event. Each new day is a new opportunity. And today, to be a part of this webinar is a golden opportunity for all of us, as we all will learn something by the end of the webinar, because it's going to be super knowledgeable and full of enlightenment. There is a request to all to fill the feedback form to get the e-certificate. There is one chance for all the students to get an additional certificate by answering a question based on today's talk, which is being asked in the feedback form. The one who will answer the best will get the additional certificate. So what are you waiting for? Grab the opportunity. The link for the feedback form will be posted soon during the lecture in the chat box. So I hope all of you will actively participate. Now, I respectfully and cordially invite Sir Dr. Deonandan Kumar, teacher in charge of Department of Chemistry, to address the gathering.
Thank you, Ms. Palak. On the behalf of the Department of Chemistry, Deshbandhu College, I welcome you all to the second lecture of International Lecture Series entitled Multifaceted Chemistry and Ever-Evolving Domain of Science. First of all, I would like to welcome our Honorable Principal, Professor Rajiv Agarwal, a wonderful teacher, a great administrator, and a humble human being. I can certainly say he has influenced many to make us feel like a one united family in the college. Under his guidance, our college is reaching new heights. The Department of Chemistry is the biggest department of Deshbandhu College. Our department is full of dedicated faculty members. They are actively participating in research and in corporate life of college. This international lecture series is initiated by Dr. Pratibha, who herself is actively involved in research. The purpose of this lecture series is to give international exposure to our undergraduate students. This will help them for their overall development. It is my great privilege to welcome today's speaker, Professor Marcelo Calderon. He is a research professor at Polymer Institute, University of Basque Country, Spain. He is going to talk about crossing biological barriers, a smart modalities to improve performance of polymer nanocarriers. Dr. Pratibha will elaborate more on Professor Marcelo's research work. I would like to invite our Honorable Principal, Professor Rajiv Agarwal, to say a few words. Over to Professor Rajiv Agarwal.
Thank you, Dr. Dev Nandan. Very good afternoon, friends, and namaskar. On behalf of Deshbandhu College, I welcome all the participants, all the students, faculty members, researchers all across the globe to the second lecture of this international lecture series, which is organized by Department of Chemistry, entitled Multifaceted Chemistry, an ever-evolving domain of science. Friends, our college, Deshbandhu College, is one of the oldest and the largest college of University of Delhi. Our college is having almost all the major disciplines of sciences, humanities, and commerce. Our college, Deshbandhu College, throughout the year is engaged in various activities, both curricular and extracurricular, other than academic activities. Our focus remains on how best we can provide a platform to the students for their overall developments. I'm sure having eminent speakers from all over the globe will help in better understanding of current developments in the field of sciences, and that young minds will immensely benefit from these lectures. I especially extend my warm welcome to Professor Marcelo Calderon, research professor at Polymer Institute, University of Basque Country, Spain. Sir, I express my gratitude to you for sparing your valuable time. Professor Marcelo will be delivering his talk on the topic, which seems very relevant and important, crossing biological barriers smart modalities to improve performances of polymer nanocarriers. The topic of the talk really interesting and fascinates me. We know that crossing our own barriers improves our performances in every aspect of lives. Being a mathematician, it would interest me immensely to know and understand how the nanocarriers cross the biological barriers. Friends, the figures, those who died in COVID-19 this year, is disturbing to the mind. But this figure could have been worse if the scientists, doctors, sweepers, teachers, leaders, actors, and citizens would not have stood with us with a determined government. Diseases like cancers, AIDS, dengue, malaria have caused great harm to the mankind. Today, we have succeeded in fighting these diseases to a great extent with the help of new inventions only. Nowadays, a new invention in the field of medicinal chemistry, polymer-based nanocarriers, is eradicating serious diseases like cancer very effectively. I'm sure we all shall be highly benefited by today's worthy lecture. In the end, I would like to congratulate the whole chemistry department, especially Dr. Dev Nandan, teacher in charge, Dr. Pratibha Kumari, program convener of this international lecture series, Dr. Indra Khan, our coordinator, DVT Star College Scheme, our dynamic IQAC coordinator, Dr. Aditya Saxena, and all the students for organizing this worthy and important lecture. So please join us in welcoming Professor Marcelo Calderon. Thank you.
Thank you, Principal Sir, for your inspiring and cheerful words. Now I respectfully and cordially invite the one who is a great mentor and the program convener, none other than Ma'am Dr. Pratibha Kumari.
प्रतिभा जी अनम्यूट कीजिए अपने आप को. प्रतिभा जी, you unmute yourself.
Is it fine now?
Yeah, it's fine now.
Okay. Yeah, thank you, Palak. A very good afternoon to all. On the behalf of Department of Chemistry, Deshbandhu College, I welcome you all to this great event. Well, I know we all are eager to listen to Professor Marcelo Calderon, but before moving to his much awaited talk, I would like to announce the award of Certificate of Appreciation to the best answer to the question in the feedback form of our previous lecture by Dr. Supriya Mahajan from USA. And the three lucky ones are, so I'm going to announce the results now for the last lecture. So the first award goes to Anant Raj from Hindu College. Second one is Santosh Kumar from Deshbandhu College. And third is Apmit Lakcha from Miranda House. So many congratulations to all the winners. Well, here you all students have another opportunity to get similar certificate of appreciation by giving the best answer to the interesting question in your feedback form based on today's lecture. So link for the feedback form will be available in the chat box during the lecture. So please don't miss this opportunity and do listen to the wonderful talk very carefully. Yeah, well, now is the time to invite to our eminent speaker. So it's my profound privilege to introduce today's speaker, Professor Marcelo Calderon, who agreed to enlighten us with his research expertise. So thank you so much, Professor Marcelo. Professor Marcelo is a renowned personality in the field of polymer-based nanomaterial. He obtained a bachelor degree in chemistry in 2003 and a PhD degree in chemical sciences in 2007 from National University of Córdoba in Argentina. During his doctoral tenure, he worked on the synthesis of polymers and visited several national and international research groups. And it's very interesting that he also visited Department of Chemistry, University of Delhi, India in 2010 for the research work. He started his professional career in Germany when he moved there in 2007 for his postdoctoral research at Free Universität Berlin, where he focused on the synthesis of polymer drug conjugates and targeted drug delivery. Then he joined the same university as junior group leader in 2010, and then he continued there from 2013 to 2018 as assistant professor. He moved to Spain in 2019 and joined Polymer Institute as a research professor. And since then, he has been working there. At Polymer Institute, he is working in the area of responsive polymer therapeutics, which holds great future prospects. His research group has developed novel therapeutic and diagnostic tool by using polymer-based nanomaterial that can sense environmental triggers and respond at the site of action. He has also worked on thermoresponsive polymer-based nanomaterial for targeted delivery. He has a big research group and various collaboration at national and international level. He has many honors and awards to his credit. So list of his credit is very vast. So I will just name a few of them. Like he received Arthur K. Doolittle Award in 2010 by American Chemical Society, Nanomet Future Grant for young scientists from the German Ministry of Science in 2012, and he also highlighted as emerging investigator by Royal Society of Chemistry. So today it would be wonderful to listen to him on the topic about the crossing biological barriers using nanocarriers. So we all are super excited to listen to you, Professor Marcelo. So I welcome you on the behalf of Department of Chemistry. Now over to you, Professor Marcelo.
Thank you very much. Thank you very much, Pratibha, for the very nice introduction. And thank you, everybody, as well, for the invitation to present our research here. So I will share now, right? This is the time, I guess. Can you all see my screen now?
Yes, sir, we can.
Okay, perfect. So I cannot see you, so I will not figure out if I get disconnected, but Pratibha, I gave you my phone number and just drop me a message in WhatsApp or give me a call. So I figured out that I'm disconnected in case it happened. Yeah. Good. So as it was already presented, maybe if you have questions then for the end and they pop up during my presentation, just write them on the chat, then we can read them and then we address them at the very end. And again, thank you very much for the opportunity to present here our work and what we are doing, or what we were doing also over the last 10 to 15 years in Germany and also in Spain. The title of my talk is Crossing Biological Barriers, Smart Modalities to Improve Performance of Nanocarriers. I will try to go slow, and I will also try to explain each of the concepts from the very basic point of view. As I understand, there are several students here, and I also heard there are mathematicians. And today my focus in this area will be especially regarding the preclinical assessment of this and which kind of assays one should consider when you want to develop a new material for nanomedicine and to bring it into the clinics. But before that, I would like to tell you a bit more about myself and about the place where I'm currently working. So this is a map of Europe, as you may have recognized, and here very much in the north on this tip is where we are working. So here, this peninsula is shared between Spain and Portugal. Portugal is right here. And right here in the limit where we are sitting, we have a few kilometers, the limit with France. So, but we have this nice environment. As you can see, there are very nice places in our small city. This is a city which approximately 180,000 inhabitants. Perhaps this for India is very small. And for me, after coming from Berlin, I also feel that it's a small city and that you can really enjoy, and it's very nice. Our institute, Polymat, is located here. So we are delocalized between 3 buildings. This is one of our main buildings, and but our labs are located here on this department. This is the Faculty of Chemistry, and we are sitting here on the 2nd floor. Okay, and about our institute was funded approximately 10 years ago as an initiative from the University of the Basque Country, which is this one here, also together with the government, and the Foundation for the Progress of the Science in the Basque Country, which is so-called Iker Basque. This foundation is in charge of hiring new personnel trying to retain talent from the Basque Country here in the area, and also to bring new talent from abroad. And they're actually paying my position currently. Our institute is divided in two sections. One section that is more related to polymer materials per se, and this is what we call here the University Institute, and most of our teachers are working in this institute. And the other one, which is the Basque Center Polymat for Macromolecular Design and Engineering, is the one where I'm working, and here we are mostly focused on science. And a little bit more about our city, you can find a very nice article in SCS Nano published in 2019, where they talk in the editorial about our city, San Sebastian, which is a city of nanoscience and technology. So here you will read, if you are interested in reading this, that this city experienced an explosion in interest on science approximately 10, 15 years ago. And since then, many institutes were established here, and also a huge number of researchers. Specifically about Polymat, we are a series of approximately 10 research groups working in a very broad range of applications of polymeric materials. So you can really see here the titles like innovative polymers. They're working a lot on batteries, for instance, and degradable materials. We have advanced multifaceted polymers, which are studying in deep the crystallization process of polymers. Then we have a series of other sort of application, and then we have our research group that is called responsive polymers for therapeutics. So here I'm the leader, but actually I'm not alone there. So regarding my research lines, we work on the synthesis of nanocarriers with different application scopes. But as I said, I'm not alone. So I have another 2 PIs that are associated to my group. And together we build this research responsive polymer therapeutic group. And one of them is Anna Belocchi. She is a doctor working at the interface between proteins and polymers. And she is focused a lot on the synthesis of stable and multifunctional biocatalysts. As well, we have another researcher, Dr. Sandra Camarero Espinosa. She's working on smart biomaterials for tissue engineering. So you can see the three together, we are covering in a broader range the applications in biomedicine of polymeric materials. If you want to see a little bit more on details of what we do, you can have a look in our homepage in Polymat. You can also check our profiles in Twitter, and you can also check our profile in YouTube, where we upload some talks that we hosted a few months ago when we had a conference about nanogels and microgels in our research group. So this is the composition of our group currently. We are approximately 20 researchers, several postdocs, several students, and PhD and master students. You can see we have a postdoc, Neha Tiwari. She comes from NCL. She did the PhD in NCL in Pune. And then she joined our group 2.5 years ago. Unfortunately, we don't have too many visitors at the moment because of these conditions on pandemics and the limitations we have in our lab space. But usually, if you have a look on my homepage from Berlin, you can see that we had approximately 40 to 50 researchers coming from all around the world over 10 years. And many of these, I would say around 10 to 12 researchers came from India, from the University of Delhi, from the Maharashtra Institute of Technology in Pune, and also from the NCL in Pune. But our group nowadays is composed by people from Colombia, for instance, from Greece, from India, Spain, of course. We have people from Argentina, Italy, France, US, China. So it's really, and Germany, of course. Oliver, I think, is in the audience. I shouldn't forget to mention him. So we have really like a very nice multidimensional and multicultural research group that unfortunately nowadays, because of these limitations on the social interactions, we cannot enjoy that much this feature from our group, but typically we try to do our best about that as well. Regarding the talk today, as I said, I will be talking about nanocarriers, and I will focus a lot on the utilization of characterization techniques to properly characterize at the preclinical level the nanocarriers that one intends to bring them into the clinical applications. So I will be talking about the motivation of our groups, a bit on introduction about what clinical trials mean and the characterization techniques that one needs for this preclinical assessment. I will use two main topics to somehow screen these characterization techniques. And one of them are about dendritic polyglycerols, which is a structure I will show you in a minute that we explored for 10 years in Berlin. and also about thermoresponsive nanogels that are also materials that we keep on exploring nowadays. And I will use this one somehow as the transversal topic, but just to focus on the characterization techniques. I will not discuss too much on detail from the scientific point of view the results, because I think this is not the space to do that. But in case you have any question about any particular topic that I discussed today, we can discuss at the end of the talk, or we can also schedule another meeting to discuss more specifically. Of course, at the end, I will close with conclusions and perspectives. Regarding the motivations of our research, so we somehow work on 4 different pillars. One of them was in the use of dendritic polymers for theragnostics. When we talk about theragnostics, this is a word that is composed by therapy and diagnostics. So there are systems where we can combine both therapy and diagnostic in one. And for this, we have used dendritic polymers in the past, which are these sort of structures that look like a tree. And here we have been able to track behavior of polymer linked with drugs and dyes, how they behave inside of cells, how they behave in certain tumoral conditions, for instance. We have as well explored a lot on thermoresponsive nanogels, which are cross-linked materials a little bit bigger than these little trees that I show you here. And in this case, one of our focus was topical drug delivery, meaning delivery of drugs on the surface of certain organs, or in the skin, for instance, or in the mucous. And here we use our materials to encapsulate different drugs and also to transport them through these layers. So we have one example, for instance, where we use them for topical vaccination, or vaccines that are applied directly on the skin. We have a lot of work done on smart drug delivery systems. And when we talk about smart in our field, it's about polymers that are able to respond to certain stimuli and to respond to this and exert a therapeutic function. Yeah, for instance, when a material goes inside of a cell, it could undergo changes on the pH, for instance, and these changes on the pH could be used to release a drug inside of the cell. And at the end, so we also explore concepts of photothermal therapy. And here we talk about concepts where we make use of light, in this case, for instance, near infrared light, which has a wavelength around 700 to 1000 nanometers. And this light is able to penetrate deep on tissues and inside of cells. And when we use certain type of materials, this light could be converted into heat. The heat could be used to destroy cancer cells, or also to trigger the release of drugs. And this is what we show in this figure here inside of the cancer cell. And these are examples of those materials that we use. But well, just a bit as an introduction, I want to show you a comparison that I find very, very funny, but also very realistic. And this is somehow, in my opinion, illustrates very well the necessity of working together with several experts from different disciplines. Yeah, and this very often we call multidisciplines, but I also like to call in-between disciplines, and I will show you now why. And this is, as I said, a very funny cartoon in a way, but I think illustrates very well how typically biologists see a cell. Yeah, so you can see the complexity that biology typically will see in a cell. And this is how very often we chemists simplify the conditions on the communication that cells have with each other. That intracellular components of cells have with each other. And I think also very funny how a physicist can also think on a cell, no, like just an element and that makes a function, but without too much of inner complexity. This is just only, of course, an exaggeration, but I think illustrates very well that different researchers with different backgrounds can have a very different view on the same object of study. And therefore, when we want to analyze and we want to explore new systems, of course, it's very fruitful when we combine the expertise and the interest from different experts. And to this, I would like to also include somehow here in between the nanomedicine expert, how a nanomedicine expert sees this. And here in these cases, we usually see a combination of both the behaviors. Yeah. So we see somehow like the biologists, we see a little complexity. We, of course, see a little bit less. And a chemist, of course, we see a bit more than a chemist. And here we can also see some biological mechanisms happen inside of cells when we are nanomedicine experts. And we try to see them depending on our expertise, more as a chemist or more as a biologist, but we see this a little bit and with more complexity than typically a chemist will see it. And why I highlight this is I highlight this because I think not only in this field, but in many, I think there is an emerge of new sort of experts nowadays. No? So experts that we are able somehow to be at the interface of the typical or classical subjects of the past, no? Like biology, chemistry, and physics. I mean, this is a very old conception. Nowadays, this is more diffuse and overlapped. And of course, there are, as I said, new experts that are popping up out of these interactions at the interfaces. And therefore, when we propose a research project and when we want to discuss new research ideas, we typically, for instance, consider our contribution when we write these projects to be the main one. No? So we think that we will develop these materials from our point of view as a chemist or a polymer chemist. But we can never forget to have collaborations with biologists, with veterinary experts, microbiologists, etc., to validate our ideas at the in vitro biological level. So when we talk about in vitro or ex vivo, we are mentioning the fact that we don't use animals. But then, of course, you also have to consider the validation in vivo, and in this case, you need to use animals. And in this validation in vivo, you always need to consider as well collaboration partners that will allow you to validate your ideas. And of course, then you work together and with them, and then you come at the end with the conclusion. And this, when we talk about transdiscipline collaborations, very often this also results in transnational collaborations, yeah, because you not often have the right expert in the next door, but sometimes you need to look into different countries or colleagues all around the world. And this is, for instance, one of our collaborations in India with the Maharajas Institute of Technology. Moving a bit ahead, a bit more into the topic, as I said, I will be talking about the preclinical assessment of polymeric materials as nanocarriers. But for this, we need to know what is the clinical development. Perhaps now with the development of new vaccines against COVID, almost everybody probably here in this audience is familiar with clinical developments or the general ideas of clinical developments. But this somehow, it's worth to be introduced anyway. So in this case, what we will be focused on is on the preclinical assessment today. And this is what typically is called phase 0 on development. Yeah, and in this phase 0, we talk about testing or synthesis at the very beginning, and then characterization at the physicochemical level, and then characterization at the biological level, that, as I said, usually is divided between in vitro and in vivo characterization. And this very often comprise studies with small animals and also with bigger animals. Then when something is clear and passes all the norms and the evaluations of a certain regulatory agencies, this can go then into phase clinical trial 1 and 2. And here, this is when typically the compounds are tested then in humans, if the medicine is meant to be used in humans at the end. And then in phase 1 and in phase 2, this is done in small groups for testing efficacy and toxicity. And then later in clinical phase 3, this is when we test our materials in a bigger group of people, then patients and also volunteers. And then later, when this goes into market, there is as well then a so-called post-market phase, or the phase number 4, where this can also be assessed over the time upon application then on the general public. But today, as I said, we will focus a lot on this area. And when we talk about materials to be developed and growing through the whole way, so typically we talk regarding time between 10 and 15 years, the whole development could take. And if we start from the scratch, yeah, from the new chemical entity, and then we play completely with the chemistry, we do characterizations at the different levels, and then we move, yeah, 10 to 15 years. And regarding money, we are talking typically here approximately $1 billion, yeah. Of course, this is a huge amount, but typically nothing is being done by one research entity or one company. Typically, one company or one research group takes care of one phase, and then they sell their idea to a next one that can take it to the next level and so on. So typically, on this first phase, you can take between 3 to 5 years on chemistry and preclinical testing. And this could take you between $1 to $5 million approximately to have an idea. So which examples have crossed already this pathway in the nanomedicine field? Well, we have several examples. And one of them that is very known, is so-called Abraxane. And the Abraxane is an injectable suspension of paclitaxel. Paclitaxel is an anti-cancer drug. And this anti-cancer drug, paclitaxel, is formulated together with albumin. Albumin is the most abundant protein in our blood, and it's actually the function that the protein has is the transport of drugs in our blood. And actually this was used and engineered in a way that it will generate a nanoparticle with this albumin with a negative charge on the surface. And then this can circulate longer in the blood and can also deliver more drug to the tumor cells. Another example is Doxil. This is as well a very known drug or nanomedicine that it encapsulates Toxorubicin is another anti-cancer drug. This is Toxorubicin here, the structure. And you can see this is it's a structure where you can as well do chemistry if you want to chemically link these to the nanocarrier. But in this case, there are not chemical linkages. There are only encapsulation of these drugs within a liposome. So a liposome is built, and because of this bigger size and extended circulation in the blood, this is what you get at the end. If you inject Toxorubicin, and this is resident time in blood, you can see that after a couple of hours, this is almost completely clean from the blood. But in case you have this drug encapsulated inside of these liposomes, after you inject them, you can see that the time is extended. And because we extended the residence time in blood, then we have greater chances that this gets accumulated into the cancer cells. Something that has to be mentioned is that the regulatory offices will always be responsible to approve the passages from these nanomedicines into the next phase. And in different countries, they have different names. The most known one around the world is the FDA, And this FDA, as well as the others, they have very well-established protocols for each of the steps that you have to pursue. So if you imagine that you would like to develop one of these systems, you would like then to have them considered from the very beginning. So then you have the right documentation when you want to cross to the next phase. Which kind of testings are typically done in each of these sections? Well, we have several, and this is just only a summary, a very general summary of what you can do in each of the phases. On the preclinical biological studies, typically you check in cells. These are typically cell lines, meaning that are immortalized cells that one can freeze in our freezer and then just store them and then use them in the lab. And animal testing to assess toxicity in vitro and in vivo, to find values about dosing, how much we can add without having any toxicity, and how much we have to add to have really pharmacological behaviors. Then we also need to study pharmacokinetics and pharmacodynamics, which this is translated into what the drug does to the body and what the body does to the drug. You need to study that in the preclinical studies. And then you somehow need to study the same things, but then in the next phases with people. You do it in smaller groups, then you do it in bigger groups and even bigger groups, and then you study a bit more in each of these phases. But it's basically the same information that you need to find at the end. You need to find information that back up the idea you have that this is a pharmacological concept that is better than the one that already exists, and that is not toxic, so that is safe at the end or biocompatible. So what do we do in preclinical phases? So here I divided the preclinical phases in 3 parts. The first part is include the synthesis and physicochemical characterization of your material. And for this, you need to establish your synthetic procedures, considering the safety of all the materials that you use, and specifically of the products you get at the end, the purification that you do with them, the functionalization you do, the chemical composition, size, shape, etc. You have to fully characterize your material in a way that you can demonstrate that this synthesis and this characterization of your material is reproducible. This is very, very important, because on the control and reproducibility of your synthesis relies then the behaviors you get in the next phases. Yeah, just to translate this in easier words, this means that you have to show that you're able to always synthesize the same material. Yeah, and that you will not get a population of different materials in a mixture at the end, because in the different population you have in this material could have different pharmaceutical behaviors and could have different toxicological profiles. So then you have to show in a very reliable way and a robust manner that you're able to synthesize this material on a reproducible phase. Then from there, you move to the in vitro biological characterization, and this could be as complex as you want. The first one that you can consider is what happened exactly at the moment I put in contact my material with biological matter. And this could be blood, this could be cells, it could be mucose or whatever. But typically what it happens, the first thing is that you create a biointerface between your material and proteins. And this is so-called protein corona. Then you also study if your material biodegrades, if you can control the release of the drugs you have encapsulated, if this is toxic, activates the immune system, if toxic at cell level, genes level, tissue level, etc. And of course, you can already show at this level therapeutic efficacy. And then ultimately, you will also check the in vivo behavior of your material. And here, then you work with animals. Yeah, and these animals, as I said, it could be very small animals like mouse, yeah, but then it can also go as big as monkey, for instance. And here you study then the tolerated dose, so how much you can inject or how much you can dose your system to these living organisms until you create any toxic effect. Even you can also consider to study the lethal effect. You study survival rate. How do you improve the therapeutics of the drugs that you are encapsulating within your nanocarriers? How the biodistribution is. And when we talk about biodistribution, is what happened to the material at the moment you put it in contact with a living organism. Or imagine if you inject your nanocarrier with the drug inside into a person, where does this drug and nanocarrier go in your body? And if it accumulates somewhere, and eventually how this is excreted as well. And today I will go through this path. Yeah, I will not consider everything because otherwise this will take until tomorrow. Yeah, very long. We have considered, I would say, almost everything in different studies, but this is a work of 10 years. Yeah, but today I will just, as I said, I will consider 2 nanocarrier systems, and I will walk you through all these 3 pathways. So let's get started then with the synthesis and physicochemical characterization. Can you all hear me well?
Can be a little louder, sir.
Yeah, okay.
Professor Marcelo, can be a little louder, please. Thank you.
Yes. So let's get started with the first compound that we use. So one of them is polyglycerol. This is a dendritic polyglycerol. It has a structure like trees. Yeah. So you can see on these trees we have branchings, and when we control this branching structure, we can also control the behavior of the material and the functionalities. You can see there are functionalities on the surface of this tree. There are functionalities in the core of these materials. There are also ether bonds in the core of these materials. Perhaps you know a very similar polymer to this one, polyethylene glycol. And polyethylene glycol is being used in many pharmaceuticals. We wrote a review about this in 2010, together with colleagues in Berlin, when I was working in the group of Rainer Hack in the Freie Universität Berlin. And we wrote this review in collaboration with Sunil Sharma. Yeah, Sunil is a professor in the Department of Chemistry, University of Delhi. Perhaps some of you already know him. And here in these materials, you can see that you have a high density of functional groups. So you have a high multivalency, and you can make use of this. This is very, very water soluble. And some components of these polymers are already approved. And several studies done by other research groups, not ours, demonstrated that the material is more biocompatible than PEG. And in this review, we explore several of the applications that can be considered for polyglycerol. And some of them, of course, include these very basic studies on compatibility of the materials. And yeah, so in this very paper on biomacromolecules in 2006, a group from Canada reported that the material has no issues when it's injected, and it can even be injected in the blood at the dose of 1 gram per kilogram. And this is a very, very high dose, for those of you know. We don't know. And when we study the half-life of these materials, when they have a molecular weight of 500,000 approximately, they have a half-life of approximately 60 hours, which means that when we inject this material, it could be probably circulating approximately 1 week. Yeah, so half-life is only for the half of the time. When it comes to synthesis, of course, you need to standardize the synthesis, and these hyperbranch polymers are typically synthesized by ring opening polymerization, and this typically starts at this point. So we have a starting point on the synthesis, we generate a base, yeah, and this base is nucleophilic, is able to attack then to a ring, yeah, so here you have a ring, and this ring opens, and then it will give you two oxygens that are reactive, and these two oxygens that are reactive then will give you the chance to synthesize a linear polymer or a branch polymer, depending on the condition you use. So when you are able to have base activity on these two systems or nucleophilic activity in both systems, then you get a hyperbranch system, which then repeats itself, the reaction all the time. No, new monomer comes, open, blah, blah, blah. And then controlling the conditions, you will reach a certain molecular weight on your material, and then of course a certain amount and also certain conditions for purification. And the patent that it was applied in 2002 about this synthesis, it reported a very well-controlled synthesis by these guys here, Rainer Hack and Stefan Mecking, that it could synthesize approximately 10 kilograms of polyglycerol in 1 week and completely purify with molecular weights ranging from 2,000 to 20,000. Polydispersity, which means the distribution of this polymer is approximately 1.6, which is acceptable, I would say. Then that was only for this structure here, so the hyperbranch system, but then we were also able to synthesize different architectures, and we actually have somebody here in the audience, Shashwat Malhotra, who was working in Berlin for a long time, as well as Shilpi is also there. And Shilpi and Shashwat were able to synthesize these sort of systems, which are so-called dendrons. No? So you can imagine if this is a tree, this will be one of the branches of the tree. And one can also build a tree by attaching together the branches, or they can also functionalize other systems to have the same behavior. We have perfect trees, imperfect trees, and also we move into next dimension and next sizes when we use them as building blocks to create bigger spheres. Yeah, and we have done a lot of work with thermoresponsive materials, but other people also working, and also other people working in the group of Rainer Hack were able to synthesize material like this one. A lot of people from Delhi University was working with Rainer, also synthesizing, for instance, antiviral nanogels. And you can see here the size ranges you get with these materials, 1 to 3 nanometers. And then you can control the sizes you want when you control the templating conditions to get bigger materials like this one. In our particular case, we work with these nanogel systems, and we use these hyperbranch systems as a cross-linker, and these are the gray spheres here. And then we connect each other through thermoresponsive polymers. Yeah, and these thermoresponsive polymers are polymers that are able to change their interaction with solvents when you change the temperature. And what we wanted to achieve here is that the material is completely soaked with water and drugs. But when we change the temperature, in our case is when we increase the temperature, these thermoresponsive materials or thermoresponsive polymers will expel the water out of the nanogel, and then water will come out as well that encapsulate the drug. Yeah, and you can see here that what we follow here is a sort of sponge effect. No, so imagine you have a sponge completely soaked with water, and then you just press it. Yeah, and this is what you can imagine very, very easily. And this is what we did. We took the sponge, we changed the temperature, and that's the effect of pressing the sponge, and then it released everything that was inside of the sponge. Regarding the chemical architecture of the materials, as I told you, we use these hyperbranch or dendritic polyglycerols as cross-linkers, yeah, these gray spheres, and this is what we had. We'd functionalize them in a way that then they could react with other monomers to create these thermoresponsive polymers. And then we have several compositions, yeah, and depending on the composition, then we had different thermal behaviors. Yeah, some of them transition at very low temperatures, some others transition at very higher temperatures, and in some cases we could fine-tune the temperature where we want to really have the transition. Yeah, of course, this takes very long time to be synthesized, and we had 3 PhD theses finished on this topic and 2 postdocs as well working in the group with the different compositions. So what did we do then afterwards? We encapsulated the therapeutic agent, and here you have just some examples of the agents we consider. Some of them like dexamethasone, doxorubicin, for instance, that I showed you before, no, this anti-cancer effect of doxorubicin. But as well, we consider peptides that have antifungal effects, asparaginase, albumin, transglutaminase, etanercept. So you can see you go from very small hydrophobic drugs to very big hydrophilic proteins, which are as well therapeutic, so they can be considered as drugs. And in these cases, we could control very well the encapsulation of the materials, and this release, like the sponge effect, we can only control for the bigger materials, no? So for the proteins. For the small ones, we have a release of drug because of diffusion and not too much because of this pressing the sponge effect that we postulate. For the Hyperbranch systems, we also have another example. I told you that when you have doxorubicin, this is the structure again, you have many points where you can do chemistry, and we basically did chemistry in this structure. So here, there was a ketone group, and this ketone group we generate in a hydrazone. And this hydrazone group is a functional group that is very stable at pH 7.4, which is a physiological pH, what we will find in the blood, for instance. But it can be easily get degraded when you go to pH lower than 6. And this pH lower than 6, it can be found, for instance, inside of cancer cells. And whenever this material is brought inside of the cancer cells to a certain compartment, this link is broken and then the drug is released. Yeah, and whenever the drug is released, it can exert the activity. And whenever the drug is coupled to this and to this linker, and in our case, through this linker to our polymer, this is behaving as a so-called prodrug. So the system is not that toxic when it circulates in the blood. It's not that toxic when it goes inside of the cells, in case that it goes to another cells different than the cancer cells. But at the moment they reach this intracellular compartment on these cancer cells, they see this lower pH, and this lower pH clips the bond and release the active drug. And this is basically what we did in our group. No? So we chemically functionalized polyglycerols. Now you see this, several of these hydroxyl groups were converted into amine groups. These amine groups there were subsequently activated to thiol groups, and these thiol groups can react with the malamido group from these prodrugs. Yeah, it's a very easy reaction that once you start this, in half an hour, you can have several grams of this compound at the end. Yeah, so then we also link a polyethylene glycol to the surfaces of these materials to modulate the size of the material in solution, to modulate solubility, and also to modulate the stability of the drug, and therefore the biocompatibility of the system. And this is regarding synthesis and regarding characterization. The typical thing you do then is to study the drug release or the protein release from the nanogels. And this you can do from several techniques. One of the typical techniques that we use is gel permeation chromatography. And this is a chromatography where we separate materials depending on their size. And here you can see this is at the very beginning, almost time zero. This is the peak from our polymer drug conjugate with a little bit of drug release. But then when we incubate this on certain conditions that it will trigger the release, you see that the peak for the drug start increasing. And this is what we expect to see. So we have a release, control because of the pH. And when we study how much is released depending on the pH we use to incubate these nanocarriers, you can see that at pH 7, there is almost no release, yeah, because I told you before that this hydrazone bond is very stable at this pH. At pH 6, there is a little bit more release, but it still is lower than 10%. But then when you go to lower pH that you expect to see inside of the cancer cells, you see that within 2 or 4 hours, more than half of the drug has been released. And then even a lower pH 4, then in 1 hour, you get 80% of the drug released and therefore active. And this we did it with GPC, yes, gel permeation chromatography. But if you also play a little bit with the chemistry, we couple a dye next to these systems, and then you can also do Fred effect, which is a quenching by fluorescence. And then you can also study the release by the recovery of this fluorescent. And then this is exactly what we did to show that the material released the drug by just only recovering the fluorescence. And that was the work done by Harald Kruger and my first PhD student in Berlin in 2015, published in Nanoscale. And the last example about characterization that I want to show you today is the release of proteins from inside of the nanocells. Here we also tracked the fluorescence of the material. And here we could see that depending on the temperature that we use for incubation and depending on the composition we have on the nanogels, these are the black names here in bold, you will have a very fast release of the protein or a very slow release of the protein. And therefore we can show that by tuning the composition of the material, we can have a tunable temperature triggered release of proteins. And that's everything about synthesis and physicochemical characterization. And now I would like to move into the in vitro biological characterization. So one of the examples that we started exploring in 2010, and that was when Jayan Kandare, who today is a professor at MIT in Maharashtra Institute of Technology in Pune, and he came with this idea of exploring the dendritic polyglycerols and a very, very basic concept and try to get a structure-activity relationship. And we did really a lot of work. We published approximately 10 papers in this field, where we studied what happened to these polymers when we start changing sizes, when we change chemical functionalities on the surface, when we change the surface charge of the materials, when we change the shape, size, etc. And the first example that we published, it was in 2010 in Biomaterials. And here we chemically modified the surface of a polyglycerol of molecular weight of 10,000, and we studied what happened to these materials. Yeah, so here now that we are in the in vitro biological characterization, we of course did some biological studies. And here, the first one that you can see is the penetration of these materials inside of cells, yeah, cancer cells in this case. And for this, we needed to chemically label the materials with a fluorescent molecule. So we linked a fluorescent molecule to these polymers. So we were able then to see by fluorescence in the microscope if the materials went inside of cells or not. And in this case, you can see in lung epithelial cells, we can see that in some cases, there are almost no penetration of the particles of these polyglycerols inside of this type of cells. And when we have the positive charges, there was some penetration, but when we have these negative charge surfaces on our polymers, we have a lot of penetration. In this case, this figure is for this one here, sulfonates. We did also a lot of other studies, and we somehow built a similar concept that this was already published in Nature Material for many other nanoparticles, solid nanoparticles. But in our case, we could also somehow correlate the same. So we study different zeta potentials, we study different rigidity on the materials, we study different hydrophobicity on the materials, and we could show that in some cases these materials are cytotoxic or they're uptaken by other cells. But what, of course, you try to determine where you have this green area that is the biocompatible area. So then you can use your nanocarrier with a safe profile to transport the drugs, yeah, because you don't want to have your material toxic for this. The next study was related to the penetration of the materials, but now regarding their size. And then we study here as well the localization inside of the cells. And these are very nice pictures you get in the fluorescent microscope when you label your polymer, but also you start labeling sub-organelles from cells, or organelles. In this case, for instance, in blue is the nucleus. And then we have our material here also spotted around the nucleus in different areas. And we found an optimal size for the penetration, the passive penetration of our materials. And when I mean about passive, it's just only going inside of the cells without any help. And I will show you now what I mean with that. And then whenever we have sizes around 10 nanometers, then we have an optimal size for this particular polymer. And now here comes the example of active. When you use a certain chemical entities, and for instance, here is a tripeptide. These three peptides are able to recognize certain receptors on cancer cells, on the surface of cancer cells, and then you get a better specificity than when you want to target them. And this is what we call actively targeting the penetration. And when you have cells that don't have these receptors on the membrane, you have almost no penetration of your materials. Yeah, you can see here. And when you have your material without these chemical peptides that are able to target these receptors, there are also almost no penetration on these conditions. But then when you have your material decorated with this peptide that is able to recognize receptor on these cancer cells, then they are being brought inside of the cells. So you see here we have very nice penetration inside of cancer cells, and this could be eventually be used to deliver anti-cancer drugs only to those cancer cells and not to the others. Regarding our conjugates with doxorubicin, here by studying as well the penetration inside of the cells and also by studying the inhibition concentrations, meaning concentrations that are able to kill these cancer cells, then here we can somehow optimize the conditions. Yeah, so we find a certain concentration in which these systems with doxorubicin are able to kill these cancer cells, and you can see that we did a correlation now with different density of drugs coupled to the polymers and also different length of the polyethylene glycos on the surface of these materials. And we use as well the concept of following the drug by Fred that we developed with Harald. And in this case, we were also able to track when exactly this drug is being cleaved inside of the cell and exactly at which location. And more specifically, a work that we haven't yet published in collaboration with Professor Alexiev. She's a physicist. Yeah, so you can see now again this trans-discipline collaboration. And here, by using fluorescent lifetime microscopy, we could show exactly the point and location where this drug is cleaved, and then how the drug moves to different compartments inside of the cell, and ultimately into the nucleus. Yeah, so what we see here is red is the drug in the nucleus, which is exactly what we expect to see, because this drug, it has the function exactly in the nucleus. And in vitro, you can as well do a bit more sophisticated studies, not only this 2D. So we call 2D because we do like a sort of surface studies on cells, but you can also do 3D studies on cells. Instead of using like a Petri dish to grow cells, you now actually use a sphere of cancer cells. And this is what is called typically tumor spheroid. In the tumor spheroids, then you have a situation closer to reality, because now you have gradients of nutrients, gradient of gases, and then you also have gradient of different type of cells. The cells here in the middle are known to be necrotic, which are most of them dead. And then you have less necrotic and more live cells when you go toward the surface. And then, of course, this is a different challenge when you want to study penetration. Before we study penetration into cells that are in 2D, and now we want to study penetration in cells at different compositions and in different organizations into the 3D object. And here we study exactly that with Gregor Nagel. We finished the thesis 2 years ago in my group. And here we could exactly see how nanocarriers are able to bring these anti-cancer drugs at different levels in these efforts, which I think it was a very nice study. And to complete the in vitro characterization, of course, you need to do a complete full assessment of toxicity. And this we did with the toxicologist of Potsdam University in Germany, the Professor Kloiser. And here, Professor Kloiser tested, with his team, of course, tested the viability of the materials in cells. So how these materials behave when you put in front of cells, whether they kill them or not, allow them to survive, how they are able to irritate the skin, the eye, mucus, etc., how you generate oxidative stress when the materials are in contact, and eventually how you as well generate certain toxicity on the gene level. Yeah, so probably you don't see that the cell die, but this cell it somehow suffers a genetic modification, and because of this modification, then later on a later generation of the cell, you will see that there is a mutation that you didn't want to have, of course. And these are only 4 examples of toxicological assessment you can do in vitro, but you can imagine that you need to do much more in order to be able to say that this is biocompatible. Now, this biocompatibility is a very complex word. And regarding efficacy, we could as well see this, how it goes with the materials inside of the skin. These are the skin cuts, how the proteins, for instance, in this case, penetrate at different levels, and then how this give you certain concentrations inside of different skin compartments. And this was a study done with Tara Hetrick in the British Columbia University in Canada. And very briefly, give me another 3 minutes to just tell you what you can do in vivo. You can imagine in vivo, it's a very complex situation. I will just very briefly give you two examples on how you can assess therapeutic efficacy and biodistribution. And one of these examples is with the dendritic polyglycerols conjugated with drugs, in this case with Toxorubicin and with paclitaxel, the two drugs that we discussed at the very beginning in the introduction. And in this case, we combined two drugs, and we wanted to study with Ronnie Sachi-Fainaro from Tel Aviv University in Israel, how the materials then interact with these drugs, if we are able to have different ratios of the drug coupled to the polymers, and then also how these materials then translate the efficacy of these two drugs combined in different ratios against different cell lines. And we studied that with her in vitro, but what it was more interesting, we studied that as well in vivo. So here, first study you typically do is tolerability, and you start injecting different concentration of your material to the animals to see at which concentration the animals start to experience toxicological effects. And then afterwards you study biodistribution, and this is what we see here. So you inject your material and then you track the material inside of the animal by different means. In our case, it was by fluorescence. And you track different organelles. Here, you, for instance, you compare accumulation in the tumors versus spleen, kidney, liver, lung, and heart, which are typically the organs that filter the nanocarriers. And you can see that there is a specific accumulation on the tumors. This is what you expect to see. And then later, you see how this tumor size increase over time. If you don't treat it, this is what you see. And then if you treat it, you see, of course, tumor inhibition. And depending on the ratios, compositions, etc., you see that some systems work better than the others. Mm-hmm. And the last example is the one we explored with Miriam Strumia from Argentina, the National University of Córdoba. Miriam is actually my PhD supervisor, and we published this paper in December 2020, where we show that whenever we have nanogels with magnetic nanoparticles that are able to transform light into heat, we can destroy cancer cells, release a drug, and this is exactly what we could see in our studies. And with this, I come to the end. I hope I was clear enough, and if not, then I will be happy to answer any question. I wanted to just give you examples on how you can go from one area to the other into this path of preclinical treatment or preclinical assessment in direction to the clinics. And for this, I would like to thank, of course, many of the group team, and I acknowledge the work of each person in almost each of the slides. I want to, of course, acknowledge the funding here from different agencies in the Basque Country, and of course, all of you for your attention, and I'm open for discussion. Thank you very much.
Thank you, sir, Professor Marcelo Calderon, for imparting such wonderful lecture. I hope all the students have captured a better understanding of this aspect of chemistry. We all are grateful to you, sir, for sparing your valuable and precious time for Deshbandhu College. I just want to give a reminder to all that the link for the feedback form is posted already in the chat box. So I again request all to fill the feedback form to get the e-certificate. Also, there is a golden chance for all the students to get the additional certificate if they give the best answer to one special question in the feedback form. Now, I would like to invite Madam Dr. Archana Malhotra, Assistant Professor, Deshbandhu College, taking charge of the question-answer session.
Thank you, Palak. Good afternoon, everyone. Thank you, Professor Marcelo, for such a wonderful lecture. Now our session is open for question answer. And I can see there are many questions coming up in the chat box. So, Professor Marcelo, I'll take up the first question. And the question is by Dr. Rajkumar. He wants to know, can we use thermoresponsive gel solid for solid tumor due to their high temperature?
Okay, this is a very interesting question. I will say that if I understand correctly, the question he or she wants to know that if the different temperature that the tumor has will be enough to trigger these changes or this transition on the nanogels. And I will say that this is very, very challenging, yeah, because it's true that because of the hyperactivity of cancer cells in the tumors, the temperature may increase a bit. This bit is actually very small, no? So I think it doesn't go even to 1 degree. And whenever we have thermoresponsive materials, we see this transition from fully swollen to fully shrunken taking a range of temperature of 3 to 5 degrees. Yeah, to have really this on-off release behavior, you need to have this jump on the temperature. This 1 degree, even less than 1 degree that you typically see on a tumor will not be enough. And for this, this is what we propose at the very end, is photothermal use. No? So we need to have a material that converts light into heat or converts any other activity into heat. So you ultimately get more degrees on the jump, on the temperature jump. Typically what is being done is a jump from 37 to 42. Yeah, so if you only want to trigger the change on the nanogel without destroying the cell, this is so-called mild hyperthermia, and this you increase approximately 5 degrees. And with this 5 degrees window, you may, if you have the right transition temperature right in the middle, then you may see differences. And then if you want to combine this change on the nanogel or the material and release of the drug plus destruction of cancer cell, then you go above, yeah, these 42 degrees. You can go to 50 degrees without having a huge effect on burning skin around or burning other tissues around. And then, of course, you can go higher values if you want. But naturally occurring so far, and I never seen anything that really allows you to have the whole transition of the material and a very, very controlled release upon this trigger.
Oh, thank you, sir. Professor Marcelo, there's another question by faculty. Dr. Ashwini Singh, he wants to know, what is the number of monomer have to be used to construct such a single nanocarrier biopolymer? Sir.
How many monomers you need? Okay, this could be as small as you want, no? Because I mentioned the work of Chilpi and Shashwat, yeah? In their case, they have a very small dendron, yeah, and probably repeating units there, you have 5. The smallest you can have is 5 or something. But then what they did, they actually build a nanocarrier by self-assembly, yeah? So they have this very small unit, they couple a hydrophobic tail to this, and then they have an amphiphile, yeah? And whenever they have this amphiphile, when you put it in water, this hydrophobic unit would like to self-assemble like a soap, no? Typical soap that we use in the kitchen at home. And this typical soap, then, if you control the condition of this aggregation and if you control the chemistry of these materials, they will have different behaviors. I mean, and they will have different sizes. You can go as small as 8 nanometers or something like this, I think Shashwat and Shippy used to get. And then you can go as big as 100, 200, 300 nanometers when you build a liposome, like the liposome that is used for Doxil, for instance. So I will say minimum, there are not really such things. So if you manage to then self-assemble your material, you can build a nanocarrier as well with a very small material.
Oh, great. Sir, I have so many questions, so I'll take up another one. And this is being asked by Dr. Poonam. She wants to know, Dr. Marcelo, which other biocompatible nanomaterial will be used suggesting for the controlled delivery of drugs?
Wow, there is a huge variety of polymers that you can use, no? So if you Google our name and you look for a review, we have also a review with Giant in Chemical Society reviews like 10 years ago or something, where we listed some of the polymers. But you can imagine the natural polymers you can use everybody, each of these ones, no? Like chitosan, hyaluronic acid, well, I think there are hundreds. I think the challenge, or the not necessarily the challenge, but the approaches that people follow are different, no? So we can divide 2 approaches nowadays, and one approach like ours, we are chemists and polymer chemists, we like to synthesize new materials, or we like to develop or to modify the existing materials to get new concepts developed, yeah? And these are probably very scientific approaches, because we imagine we have to go almost from the scratch in this preclinical development to the full development. So we need $1 billion basically.
Okay.
But the other approach that you can follow is more the pharmaceutical approach, or the pharmacotechnology approach, where people will use polymers that are already approved by the FDA or the EMA or the Indian regulatory agencies. And then you use these materials that are already approved, and then you use them to build a nanocarrier and then deliver. And with this, you can shortcut this 10, 15 years development you need to do. Of course, you do much less chemistry. And for me, that's not that funny. But I still approach that nowadays because I'm also shifting my interest toward the translational or development of materials for real clinical use. So you can have these two, and if you go, for instance, to any pharmacopoeia, or you go for sure to your agency, and for sure they have a book. Typically, these guys, the FDA, the Indian Regulatory Office, the European Medicine Regulatory Agencies, all of them, they have the book. It's typically called pharmacopoeia, and in these books you can have a look which polymers are already approved, and there you will find a list of at least, I don't know how many, but for sure 50 or 100 probably. Yeah.
Wow.
Yeah. Polyglutamic acid, for instance. PLGA or polylactic acid. Polylactic acid is used since long. Polyethylene glycol. Yeah, there are 100 probably.
Professor Marcelo, there's another question by Dakshina. She wants to know what makes nanomedicine to be specific to the target.
That's a very good question. I think I typically give a lecture that it takes 6 months to explain that, to answer that question. But I think the very unique feature is that you can have or that you can fine tune the sizes. No, typically if you have a small drug, the typical small drug has a size in solution of 1 to 3 nanometers maximum. Then if you imagine that you want to target a cancer cell, you have a cancer cell that has approximately 1,000 nanometers, or more. I mean, this is 1 micrometer, maybe 100 micrometer, so 100,000 nanometers. You have this size difference. And because of that, this drug, it can hit that one, but it can hit many, many others because they don't distinguish, So then when we have our nanogel with sizes in the middle and with shapes in the middle and with charges different in the middle and with chemical decorations that can only recognize that cell, then you can now understand that this really plays like a transport, So you have a specific transport. You have your how you call it? Your taxi, your motor taxi that brings you exactly only to the cell you want, if you're able to build and to give the instructions to this taxi, And I think this is the unique feature that nanocarriers will have in compared to many other technologies. And also, for instance, nowadays also virus capsids are used for this, And the virus capsids could have certain biological toxicity issues that these materials will not have. Yeah, but of course you need to evaluate what you want really at the end of your material. But I would say this is a unique feature compared to the others.
Great. We have an enthusiastic audience, sir. Another question by Dr. Shikha Shrivastava. She wants to know, what is the efficacy rate in terms of bioavailability of these nanoparticles in comparison to non-nanoparticle based drugs?
Yeah, okay. This is also, I think, another semester answer, because this is more in pharmacology, But I will say that there is not a general answer to that. It's always related more to the bioavailability, means how much of this drug, for those of you who don't understand the term, is how much of the drug you apply ends up in the blood, And in principle, the values could be the same for small drugs and for big drugs. I think the main difference then it comes from the pharmacological point of view on how much of this drug you can really bring to the target tissue. The typical example is, for instance, for anti-cancer drugs. So if you imagine you inject an anti-cancer drug, this anti-cancer drug is typically hydrophobic. Yeah, it's a small drug, non-specific. And at the moment you inject it on your bloodstream, it will go to the heart, right? From the heart to the lungs, from the lungs to the heart again, and from the heart it goes to the whole body. And what is said that in 5 minutes, this anti-cancer, very toxic drug or any other drug, it has already touched almost every single cell in our body, in 5 minutes. This is, of course, a problem because you don't want that this toxic drug touch every cell in our body. But on the other side, it's also a specificity problem because it will not be able to really accumulate in the cancer cell that you want. And typically what you get is 0.01% of this drug really accumulated in the tumor. And it's already out of the body in probably half an hour. Yeah. And with your polymer, what you can reach is typically 10 folds to 100 folds, meaning 0.1% of this drug can be then accumulated on the tumor to 1% approx. Yeah. So you can see still there is a lot of room for improvement. Yeah, so there is really a lot of work that needs to be done by all of us here, researchers and scientists and pharmacists, etc., to be able to develop these materials that then can improve these values. Nowadays, there are some studies where they show that you could bring up to 10% of this drug to the cancer, but these are really the top winners, and there are only few materials that can do that. So we can still improve 10 to 100 times, but we can still improve another 100 times. You know, this is somehow the situation nowadays.
Yeah. Another good question. It's by Deepak Ranjan. Is any other serious disease can be diagnosed by using nanocarriers polymers?
Yeah, I will give you an example then from Yayant. No, Yayant Kandare. I don't know if Yayant is here, probably not. There is a very interesting topic that he's exploring in his university and also in his company. He has a company, Onco Target, I think is the name. And what they diagnose in this case, nowadays, that there is only one company, or there was, now there is an Indian company, but there was only one company in the world that was able to count, for instance, circulating tumor cells. What is the relevance of this? The relevance is that typically 90 to 95% of the cancer patients die because of the circulation of the tumor cells in the blood, that they go to other organs and they create secondary tumors. Yeah, what is typically known as metastasis. Yeah, very often the primary tumor, imagine you have the typical case, the breast cancer, very often can metastasize and move cells from the breast to the lungs. They circulate and then they go to the lung, and then you typically get treated because of breast cancer. Very often the treatment is very successful, but later, if you really have circulation of these cancer cells in your blood, they went to the lung, you will later, much later, will know this. Yeah, and typically when patient knows this issue, it's already too late for treatment. And Professor Kandare, he developed a material that is able to extract these cancer cells, which are typically 1, 2, 3 cancer cells in millions of other cells in our blood. And then he was able to extract these cancer cells from the blood and to quantify them. So now imagine you undergo this surgery, and then to know whether you need to continue your treatment to destroy the cancer cells that probably are already in your lungs, you need to count how many of these cells you have circulating in your blood. Yeah, and this is very, very important because this will enhance a lot your survival time. And this is, for instance, something that is uniquely performed by nanomaterials. Yeah, so what he does is so-called liquid biopsy. You take blood from you, and from this blood, then they use this nanotechnology with iron nanoparticles, which are magnetic. These magnetic nanoparticles are decorated with other materials and also with certain antibodies that are able to recognize and separate only these cancer cells from the blood. And then he separates them, quantify, and he can tell you whether you should continue with your treatment or not. And this is a work that he developed since 5, 6, 7 years in the lab. Later, he moved into the clinics, and to the clinic, he did it with the Tata Hospital in different locations in India. And then he later got the approval. And nowadays, this material is approved in India to be used.
Huge work, sir. Huge work.
Fantastic. Congratulations to Yayant. Yeah.
Yes. Another question, sir, by Shruti Goyal. She wants to know what are the disadvantages of using nanoparticles in drug designing, if any, what?
Yeah, of course there are. No, I mean there are pros and cons on everything we develop. Yeah. I think, of course, I just show you a tip of the iceberg of one of the issues, and the issues could be as well toxicology issues. Yeah, because at the moment in the case, for instance, of circulation, I told you, we develop a material that then can go to the cancer cells. They can circulate longer, and they can go to cancer cells. But when they circulate longer, they can also get accumulated in the organs that are responsible for filtration of our blood. Yeah, for instance, like liver or spleen or lungs or kidneys. So one of the potential problem is that we now create a new type of toxicology, which is so-called nanotoxicology. And this is, of course, something that needs to be explored in our body. And of course, you also need to consider the environmental issues, because now we create a new sort of nanoparticle that then needs to be biodegraded in the soil, in the seawater, and lakes and so on. No? So we also created potential new issues that need to be as well considered.
Oh, thank you, thank you. Another last question, I believe. Though there are many, sir, but I'm sorry, but I mean, I'll take this one. Atulya wants to know, are these polymer nanocarriers diagnostic and therapeutic currently affordable for normal people?
Yeah, the examples I gave at the very beginning, doxil, no, so doxorubicin inside of liposomes, and the other one was Abraxane, no, paclitaxel albumin. Those are affordable. These are in most of the hospitals, and these are, of course, more expensive than the drugs themselves. But they are then evaluated whether they are worth to be used on the patients or not, and they are relatively affordable. I think the question really is, as well, is my answer only talks about the tip of an iceberg, because at the moment we start talking about personalized medicine, like in this case is when we start to use materials with a very, very high specificity for a certain disease. I didn't mention, but this should also consider the specificity for the patient condition, yeah, and the patient physiological profiling. And then we probably need to, in the future, to personalize each of the treatments. And of course, this will enhance the costs. And there is a huge discussion how this will be afforded in the future, no? If the health insurance will be able to cover these things or not. Of course, the effort has to be done in both directions, to develop fancy new technologies, but also to develop affordable fancy new technologies. Yeah? And we should never forget that.
Oh. Thank you. So I think, I've taken most of the question, but, one question I think been put up in the chat box, and I'm just taking the essence of that. And it's that, this question which is based upon that compresses many questions. And it's basically asks, how is it integrating academics with industry?
Okay.
Yeah, that's, I think, yeah.
General,
yeah.
I think every country does it differently. No, I can see the examples of Germany. I can see the example of Spain and Argentina, which I know very well. I think in all cases, the most important player. Well, this is probably my view only from my limited experience. No, but in all cases, I think a very important player is the state. Yeah, because the state should fund programs that involve both players. Yeah. And as I say this because, for instance, I know how science and research and development works in Germany, where it's a very, very powerful country, economically stable, and so on. And many big industries in chemistry are located there, and they do research there. And it's a typical example that then they have also production units in different countries, like in Argentina, but then they don't have ID there, no? And they don't have research and development there. And probably states should be more active into involving or pushing these companies to fund local research groups and to work in collaboration with them. Because of course, if you talk about cancer, which is a general disease for the whole world, and then you imagine, okay, it's fine if they do this sort of research in Germany, that it will be useful for the whole world. But if we talk about, for instance, Chagas disease, or if we talk about malaria, or if we talk about diseases that are more local, then it will make sense that these companies are pushing a way to get involved with research groups in the near proximity, and also to follow therapeutic targets that are only important for those people, which very often are known as neglected diseases. You know, these Chagas and I know yellow fever and things like this, because they're only happening in small populations, poor, typically developing countries, which do not represent a commercial target for these big companies. And perhaps the state should be active into pushing companies to locally invest on those things. And how it's really going nowadays, I will say today I somehow show you an example of that, because when we move from preclinical assessment to clinics, I told you that you have these stages, and in these stages you multiply the money you need by 10. So first study in preclinical, typically done in universities. For that you need a couple of millions, but then for the phase 1 you need 20 millions, for the next one you need 200, and for the next one you need 1,000. And then when you need to jump, you typically need to jump by moving into your ideas into a company that is bigger than the one that was developed in the former one. Yeah. So typically I will say in these developments, the relationship between companies and industry are in the preclinical jump into the phase 1 or jump into the phase 2. There you are obliged to interact with a company that then brings this forward. Of course, you need to convince them to pay for that. You need to, the typical numbers you get is if you sell, typical, no, it's give and take. The typical number, if you have a whole preclinical assessment done, if you build then a spin-off company in your university with that, you could probably sell your material and your idea and your proof of concept of preclinical study of phase 1 for 10 to 20 million euros or dollars approximately. Yeah. And there is where you need to interact. You have no more option unless you get 100 million euros from somewhere, which is very difficult to get it from governmental institution.
Oh, great, great. Thank you. Thank you so much, Professor Marcelo. I mean, we are really honored to have you with us. I mean, your lecture was totally captivating. Isn't it, Dr. Pratibha?
Yeah, really. The lecture was really very interesting, very informative. We enjoyed, we learned so much from your lecture. Like, I would like to ask one thing, like, there is so much about nanogels, nanocapsule, nanosponges. So, now, what's the basic difference and which one is like having more efficacy toward like drug delivery and all this? What's the basic difference and what should be the basic functional group which allow the crossing of the biological barriers? Like, which type of material you think is better to like pursue for further studies?
Again, another question that it will take me 6 months to answer. No, I think it depends a lot on the application. Yeah, I like to think that the nano gels are somehow more general because you can imagine the nanogels to be much bigger than the others. No, the nanogel is approximately 50 times bigger than the small dendritic units. And then if you build your nanogel and that is fully degradable, then you can have this sort of idea bigger that then chops into smaller pieces that can do the rest of the work. No? For instance, the work I show you where we study the tumors in 3D, there we pursue exactly this idea. So we have a bigger size, approximately 100 nanometers on these nanogels that are able to circulate and accumulate very well on the tumors. But because of this big size, they cannot penetrate deep on these spheroids. Yeah, and for the bigger penetration in there, you need to have your material fully degradable into the smaller active units. And this is what we did there. So we have the material that can accumulate on the area where the tumor is located. And then because of the expression of certain enzymes by this tumor, the material can get broken to smaller pieces. And then these smaller pieces can go deeper areas and kill the deep cancer cells because of smaller sizes and a certain functionality. And I think this will be one smart way to go, I think. Somehow making use of both ideas, yeah, but starting with the nanogel. But again, this depends a lot on the application. That's only one example in solid tumors, but you can imagine that there are many other ideas that you can explore. If you want to have a big gel, maybe these nano things are too small, and then you need to have a more cross-link and bigger material. But well, yeah. As I said, we can set up a course for 6 months and then we talk.
Yeah. So we hope we can have another lecture.
Yes,
and we can explain few aspects like which type of material is better.
Yeah. So imagine in the future you find a lot of interest on your students saying, okay, I would like to learn more about mucosal penetration that I didn't talk about today. We can set up a whole lecture on that and then see which targets. And because in mucus, of course, you have mucus all around the body, and then each mucus will have different composition and will need different type of nanocarriers. And this is, of course, as I said, a 6-month lecture.
Yeah. Okay. Yeah. Thank you so much. Yeah.
Thank you all.
Thank you, really. That's so right, Pratibhaji. I mean, we are so fortunate and that you humbly accept our invitation. Thank you, Professor Marcelo. I mean, we're really, on the behalf of department, I would like to thank you for your wonderful lecture. And we'd like to see you again. Once again, a big thank you, Professor Marcelo. Over to Palak.
Thank you, everybody. Have a nice rest of the day.
Thank you so much. Yeah.
Bye-bye.
Yeah, over to Palak.
Thank you, sir, Professor Marcelo Calderon, and thank you, Dr. Archana Malhotra, for handling engrossing question-answer session. Now, I would like to invite Madam Dr. Seema Lal, Assistant Professor, to give the formal vote of thanks.
Hello, good evening, everyone. Am I audible?
Yeah.
Yes, you are.
Thank you, Palak, for giving me honor for making closing remarks on today's wonderful webinar. On behalf of the Department of Chemistry, Deshbandhu College, I am taking this opportunity to extend my sincere thanks to Professor Marcelo from Polymer Institute, Spain, who spared time from his busiest schedule and delivered such an interesting and informative talk on polymer nanocarriers and their development in the area of smart drug delivery and ability to cross the biological barrier. Today, we all had an opportunity to hear about his fascinating and instructive research, and this will surely be going to encourage our students in their future events. Thank you, Professor Marcelo. Thank you so much. I would like to thank our dynamic and energetic principal, sir, Professor Rajiv Agrawal, under whom guidance our institution is flourishing day by day. Thank you, sir, for your consent and support throughout this webinar and the lecture series. Further, I am very thankful to Dr. I.K. Singh, sir, Coordinator, DBT Star College Scheme, Dr. Aditya Saxena, sir, Coordinator, IQAC, Dr. Devanandan, sir, Teacher in Charge, Department of Chemistry, and Dr. Pratibha Kumari, ma'am, Program Coordinator, and entire fraternity of Department of Chemistry for making this webinar a successful event. A very special thanks to our student team. Palak, can you please show their names? Yeah.
Yeah, it's already there.
Yeah, it's already there.
Okay.
Yeah.
Thank you, students, for putting lots of effort in organizing this event. Thanks a lot. In the last but not least, I'm thankful to all the participants for attending this enlightening webinar. Thank you, everyone, for being with us. Thank you so much. Have a good day ahead. Over to you, Palak.
Yeah, Palak.
Yeah, ma'am. Thank you, ma'am, for your kind words. Now I hope that all of you have filled the feedback form and have participated actively. Remember, do follow the Deshbandhu YouTube channel and join the Telegram channel as well to get the information for more events of International Lecture Series. Hope to see you soon. Stay safe, stay healthy, and stay home. Thank you so much for joining us. Have a delightful and marvelous day.